Here we demonstrate methods for measuring expansin activity by cell wall extensometry. Suitable wall specimens can be prepared from the hypocotyls of etiolated cucumber seedlings. To measure acid-induced extension, which is catalyzed by endogenous expansins bound to native cell walls, we clamp the walls at neutral buffer and an extensometer. Upon switch to acidic pH, expansins are activated and the cell walls extend rapidly. We also demonstrate an expansin reconstitution assay. For this part, we use a brief heat treatment to denature endogenous expansins. Heat-inactivated cell walls do not extend even in acidic buffer, but addition of expansin rapidly restores wall extensibility.
Hi, I'm Dan Durachko from the laboratory of Dr. Daniel Cosgrove in the biology department at Penn State University. Today, we will show you a procedure for measuring irreversible extension, or creep, of isolated plant cell walls. So, let's get started.
In our experience, young hypocotyls from etiolated cucumber seedlings serve as a convenient source of cell wall material for these experiments. Cucumber seeds are sown on wet paper in a light-proof box, which is kept in a darkened cabinet in a constant temperature room set at 26 degrees Celsius. Temperatures between 22 and 30 degrees Celsius will also work, but it is important to note that the temperature will determine the rate of growth. The warmer the temperature, the faster the seedlings will develop. Maintaining a dark growth environment is critical, as even small amounts of light affect both the rate of seedling development and the cell wall properties that we measure with this technique.